Bacteriophages are among the most numerous microinhabitants on Earth. Imagine just 1 liter of seawater contains around 10 billion bioparticles, while a single gram of marine sediment can house millions to billions of these organisms. Oceanic bacteriophages play a crucial role in controlling bacterial populations and act as massive reservoir for essential elements like nitrogen and phosphorus. These bacterial viruses are also key natural transmitters of genetic information across the world's oceans, ensuring the annual transfer of staggering amount of genetic materials. Their influence extends far beyond simple infection. They shape ecosystems and drive evolutionary processes. Understanding their abundance sets the stage for exploring their classification and taxonomy, which helps us make sense of their diversity and function. Classifying bacteriophages is essential for several reasons. It enables scientists to identify new phases from natural sources, maintain organized collections and databases, and detect viruses that cause economic losses in biotechnological processes. Classification also aids in enforcing strict propagation control, identifying bacterial viruses for phagotyping pathogens in medical studies and using them as therapeutic agents. Moreover, it optimizes training programs in microbiology and biotechnology and helps decipher phylogenetic relationship between individual phases. By understanding how bacteriophages are categorized, researchers can better track their evolution, harness their potential in medicine, and manage their impact on industries. This systematic approach is the foundation for all further studies and applications involving bacteriophages. The classification of bacteriophages has evolved over time. Bradley's scheme divides phases into 6 morphological groups based on their structure and nucleic acid type, from long-tailed phases with contractile or rigid sit to short-tailed grand-site nucleocapsids, single nucleocapsids, and lord-like filamentous phases. Notably, Group E phases contain RNA, while other mainly have DNA. Tikhonenko later merged some groups, creating 5 main morphological types. The International Committee on Taxonomy of Viruses (ICTV) now oversees the classification, with the current scheme consisting of 1 order, 13 families, and 34 genera. Families are distinguished by nucleic acid type and variant morphology, while using over 40 criteria for differentiation. This structured approach helps scientists understand phases, diversity, and their evolution relationship. Tailed bacteriophages, classified under the order Podoviridae, are the most numerous and widely distributed group of bacterial viruses. Their capsids are made of protein coats and liner double-stranded DNA with distinctive cube-shaped heads and spiral-shaped tails. These tails often end with specialized elements that help the phase attach to bacterial cells. Podoviridae are divided into 3 families based on tail structure: Myoviridae with long contractile tails, Siphoviridae with long non-contractile tails, and Podoviridae with long rigid tails. Each family has unique structural and genetic features, occurrences, and phycological characteristics. For example, Myoviridae includes the well-studied T4 phase, Siphoviridae includes lambda and T5, and Podoviridae includes T7. This diversity highlights the complexity and adaptability of tailed phases. Non-tailed bacteriophages make up a smaller portion of classified viruses, grouped into 10 families and 3 main morphological types: polyhedral capsids, filamentous forms, and diverse shapes without clear symmetry. Some families like Microbiology, Corticoviridae, and Tectiviridae have unique features such as lipid envelopes or vesicles. DNA-containing phases with polyhedral capsids infects a wide range of bacteria, while RNA-containing phases like Myoviridae and Siphoviridae target specific hosts and have distinctive genetic mechanism. Filamentous phases, such as those in the Introviridae family, are flexible or stiff rods, infecting various bacteria and showing resistant to environmental stress. Pleomorphic phases like Plasmaviridae and Fusiloviridae have unusual structures and infect specialized hosts. This diversity illustrates the remarkable adaptability and specialization of bacteriophages. In summary, bacteriophage taxonomy and classification have evolved from simple morphological schemes to complex, multi-criteria system overseen by international committees. Today, classification relies on nucleic acid type, variant morphology, and over 40 specific criteria, resulting in 1 order, 13 families, and 34 genera. This structured taxonomy enables researchers to identify new phases, understand their evolution relationships, and harness their potential in medicine, biotechnology, and environmental management. The ongoing efforts of the International Committee on Taxonomy of Viruses ensure that our understanding of phases diversity continues to expand, paving the way for new discoveries and applications. Bacteriophages remains a fascinating and vital part of the microbial world, shaping life on Earth in ways we are only beginning to fully appreciate. Thank you for watching. Stay tuned. Bye-bye.